Thank you, Manfred. It's a pleasure to be here. Can you all hear me at the back? Good. So I'll tell you a bit about genetics, to put it in context, what we mean by epigenetics, and then we'll move on to some examples of epigenetics and how it's having an impact on society. And epigenetics is currently very popular. You can see it in the popular media, whether in a more conservative type of depiction here or your German version here. And you also see it in science magazines as well. And why is it? We'll come to. And epi means above or on top of, so epigenetics literally means above genetics, and in that sense, it literally translates to the German Ubergenetics with a umlaut, right? On the other hand, Uber is now kind of moved into the English language as well, without the umlaut, and there it's a slightly different meaning, and this is the definition I fished out from the internet. Of how it's a kind of an extreme or special case, and you'll see there's also aspects of that in epigenetics. So that's why the talk title does not have an umlaut. It's both the German and English version of the word. And basically epigenetics comes down to this age-old question of how much we are shaped by our nature and how much the environment shapes us. And in biology, more specifically, we call this the genotype. So that's the genetic material that we carry and we then transmit to our progeny. And meanwhile, there's the phenotype, the actual characteristics we have in our lifetime, which to a large extent are shaped by the genes we carry, but can also arise through environmental influence too. And in fact, Aristotle was the one who coined the word epigenesis more than 2,000 years ago. And back then, he was quite ahead of his time in realizing this distinction that there's a genesis and then there's an epi part that kind of influences your life afterwards. And it's quite interesting to go back and read some of the things he said back then. So he understood that the male genetic material came from the sperm, and how it was kind of a concentrated life form, and that's why apparently males usually feel exhausted and weak after sexual intercourse, if you read the paragraph. And then he went on to discuss what about the female equivalent, and what does a female contribute in terms of genetic material? And there's also the question asked whether you should derive pleasure from sex for it to be important in the genetic transmission. And he even goes on to discuss kind of the female orgasm and female ejaculation, which for some reason he says is more common in fair-skinned women who are typically feminine. Anyhow, for about 2,000 years, this word epigenetics was kind of slumbering and forgotten. And I think I am using it. Can you not hear me? Is that better? Okay, I'll stand closer to it. So this word was kind of slumbering, and I think people here might not realize that it's partly due to Freiburg that epigenetics has had this rebirth. And that's because about a century ago, to try and distinguish between these different mechanisms, Haeckel, a biologist, came up with this other term, phenogenetics, which is kind of disappeared now. And he was also trying to describe this nature versus environment balance. And because of that, and before I go on, I should say, so Haeckel was a reader here in the University of Freiburg and then moved on to become a professor in Stuttgart, so very much a local person. And due to this history, 2 decades later in the UK, Conrad Waddington, who recreated this word epigenetics because he wanted to dissociate himself and come up with a new word. And he chose to use this old word from Aristotle that had been forgotten. And if you look at Waddington's definition, it's quite vague. It really just says something about genes and products, which is not very clearly defined. And this also was not widely used or had much currency until the 1990s, when we started to understand better the mechanisms of how genes work and how they are regulated. And Holliday, particularly in the UK, started using the word epigenetics, and now you see a more clear definition And that's how this is the mechanisms by which you're determining what genes are turned off where and also when. And if you look in Wikipedia now, the definition's at the bottom of the slide, and it's an even more slightly precise definition where it deliberately says that these are not involving the genetic code, but other mechanisms which can be transmitted. So I need to tell you a bit about the genetic code as a context for that. And this is a question that life needed to grapple with. You need to encode information, and what kind of language do you choose? The simplest possible language would have only a single letter, but that's not a very efficient way of transmitting or storing information, as you can see. And once you get to a 2-letter alphabet, for a 10-letter word, you already get many more possibilities. If you get to a 3-letter alphabet, it increases exponentially. And life actually uses this 4-letter alphabet. It doesn't have to be the only solution, of course, but very early on, once this started to be done in the primordial soup, any other alternative way of encoding information would have rapidly died out because it wouldn't have been able to withstand the selection pressure. But at the same time, we all recognize this binary language as perhaps the simplest, efficient way of storing and transferring information, and that's the way computers work. And it's also the way we feel others would understand information. So, in fact, when the human species beamed this message out to outer space, to the globular star cluster, this was a binary code message. And we don't know whether it's been received because it's going to take 25,000 years to get there. But the message itself is based off 1679 bytes, and it was believed that any civilization advanced enough to receive this message would understand that 1679 can be expressed only as the product of two prime numbers, 23 and 79. And if you arrange this message as 23 times 79, then it would mean nothing. But if you arranged it as 73 times 23, then you can start seeing patterns in these zeros and ones, and you can see them more clearly if I color code them. And in fact, you'll see much of this message is related to the genetic code. So the message tells these other civilizations how DNA is made. And that goes back to DNA, which also has local context. So DNA, the nucleic acids were first discovered by this biologist Misha, who was working in Tübingen. So again, the state of Baden-Württemberg has a big influence on genetics and epigenetics. And about 100 years later, that's when Watson and Crick came up with the familiar double helix model for its structure. And about 50 years later, the human genome was completely sequenced. And there were some surprises from that, which I'll come to in the next few slides. But meanwhile, in terms of storing information, almost any set of chemicals will be able to do this. You can use chemicals and read their message. That's not a problem. However, you also need for life some means for efficient self-replication, and that's extremely difficult to engineer. And the DNA does this very well. It has these simple rules that when you have an adenine, it always pairs with a thymine, and similarly when you have a cytosine, it always pairs with guanine. So this gives you a mechanism for creating copies of yourself in high fidelity, and that's necessary for life. And also these sugars are not flat, and when you line these up, that's why they twist up into the familiar double helix. And the way life works then is you have this information that's coded by the DNA in our genes, and this DNA is read by this protein called RNA polymerase, which makes a copy of this, which is almost looks like DNA, but it's a much less stable species. And that's so that the message will not be permanent. And this message then moves out into the cytoplasm of the cell, and that then leads to production of proteins, which is what really life is. So we are all about 15% protein. And in terms of these languages, if you like, these are both 4-letter alphabets, whereas this is a 20-letter alphabet, and so there's a difference between the two. And so How do you do that? If you have this 4-letter alphabet, if you have a word length of only 1, that only gives you 4 combinations. A 2-letter word gives you 16, so these are still less than 20 that you need. Whereas a 3-letter word already gives you 64, which is more than you need. But that is good because it gives you built-in redundancy into the system. And this is in fact how the genetic code works. You have these 3-letter words, 64 of them, and each of them has a precise meaning, coding for either an amino acid, or 3 of them also code for stop signs as well. And this is important because once in a while there are going to be mistakes in the genetic replication, and you don't want all of them to be harmful. And these amino acids are coded according to similarity. So for example, if you look at UUU, that codes for the amino acid phenylalanine. If there's a small mistake and it becomes UUC, it still codes for phenylalanine. And if you go from UUU to UUA, you're changing from phenylalanine to leucine, but those two are not that different in properties. So that's a relatively benign mutation. But there are also cases you'll see where you can move from one type of amino acid to a different one. Sometimes this can be beneficial. Sometimes this can lead to disease or other problems as well. But this is all necessary for evolution because this gives you a chance to sample diversity. So here's one of the surprises from the genome project. Before the sequencing was done, it was assumed that somehow the number of genes will scale with complexity, and the more complex the organism was, the more genes you would contain. And here you see the budding yeast and 2 parasitic organisms. So these are all microscopic. And you can see even here there's a variation in genome size. And if you look in the plant and animal kingdom, you see the same thing. So in fact, Donald Trump has roughly the same number of genes as the German cockroach. But before you get excited, all of you also have the same number of genes as a German cockroach, okay? And meanwhile, the rice plant has roughly twice the number of genes as ourselves. And another big surprise that came out from genome sequencing is that most of our DNA does not actually code for a message directly. In fact, only about 1.5% of our genome directly codes for protein. What do the rest of these 98% do? Well, for a lot of them, we are ignorant. And when I was a student, we used to call it junk DNA. But clearly, they are not junk. They're like the dark matter in the universe. They are there, they are doing something, and we largely do not know yet what they do. And then the idea was, well, maybe it's not the size of genes, number of genes that's important, but the genome size. And does that scale with complexity? Well, the answer is no, again. So if you look at the human genome size, it's up there, but you can see there are reptiles, plants, and even the amoeba has a larger genome size than humans, okay? But somehow it turns out that the ratio between the two is very important, and that does scale with complexity. So if you look at how much non-coding DNA you have as a ratio of the genome, you'll see humans and higher organisms have the highest ratio. So that clearly tells us that these non-coding portions of the genome are not junk. They are there for a very good reason. We may not understand it yet, but they have important functions. So let's quickly, probably this most of you will be familiar with anyhow, tell you how genes do work in terms of transmission. And this could be understood by observations of phenotype without really knowing what DNA was or how it looked like or what it might do. And this was done by Mendel and many others. And just to give you a simple example, this is a snapdragon flower. So the gene comes from this the protein that's coded by this gene called R, capital R. So if you have 2 copies of R, that gives you a red color. If you have 1 copy of R, that gives you a pink color. And you have no capital R's, that gives you no production of that protein, so you get a white flower. So if you cross a white and a red, you always get 1 copy from this, which is the capital R, and 1 from here, and that gives you the pink coloration in the first generation. And if you cross between them, you can then recapitulate all these earlier phenotypes as well. So you can also get statistically RR, big R, small r, and 2 R's too. And this is a scientific explanation of how, when people say characteristics skip a generation and how often you look more like your grandparents in behavior. And this is true. This is how genetics works. But in addition to that, for evolution to work, you need a variation in genes so that you can sample and select for the fittest. And this happens because the DNA replication is not perfect. Once in a while, there will be a mistake made. And it also happens because there's always exposure to DNA damaging agents like UV radiation or chemicals. So this will give rise to gradual changes. If these take place in a somatic cell, i.e. in a non-reproductive cell, then that can change our phenotype. If they take place in the sperm or the egg, then they will be transmitted to the next generation, this change in DNA. But in addition to that, for organisms like us that have sex, then that's a wonderful way to exchange genetic material, because it means with every new generation, you're taking out half the genome and replacing it with another genome. So that's the biological importance of sex. And here's how evolution might work. So you see these giraffes, which are perhaps in a time of famine. And these giraffes, their height, just like us, should follow some kind of statistical distribution. And the idea is that the taller giraffes will live longer in these times of famine and reproduce, and their genes will then be transmitted to their offspring. And over time, in successive generations, the average height of these giraffes will increase. So that would be the classical model to explain this phenomenon. And we'll come back to another one later. But before that, here's a paradox for you. In physics, you have the Schrödinger's cat, which is simultaneously dead or alive. So I can propose to you an epigenetic cat, which is less gruesome, and you need to decide whether it's male or female. So this sounds like a 50-50 question, but it's not, as we'll see later. So here's an alternative, controversial explanation for these giraffes that was proposed by Lamarck and was basically abused, and he never had acceptance of this during his time. And this is the idea that these giraffes that stretch their necks to reach these leaves live longer and reproduce. That's fine. But he also implied that this stretching behavior, which is not genetically encoded, would be somehow transmitted to the next generation. So that was the controversial part. And what he was really saying is that this stretching phenotype was being inherited without a change in genotype. So in that sense, he was ahead of his time, because this is exactly how we define epigenetics now. And of course, he did not understand the ways it could be achieved that we do now. And we'll come to this now. And it turns out that in cells, DNA is not naked in its double helix. It's always wrapped around these proteins that are called histone proteins. And the interactions between this DNA and these proteins are what determines whether genes will be read or not read. And I personally, as an operational definition for epigenetics, this is what I would use. And epigenetics is basically the science of how these changes in the nucleic acid and the histone proteins control whether a gene is being turned on or off. And in this definition, I'm not saying anything about whether that's heritable or not. That's a controversial topic, and at the moment, we don't have a huge amount of evidence that it can be herited, but these changes are certainly taking place in us all the time, and they have important consequences. And part of the reason for these changes is that the RNA polymerase that reads DNA cannot do it by itself. It needs to be brought to the gene by all these associated helper proteins. So it's a very complex process. And the way this is orchestrated is that DNA in cells is not a frozen structure. So we have cytosine residues, these Cs, and some of them are being converted chemically to another residue called methylcytosine. And this has a big effect on genes. So when this happens, it then silences a gene. So it's not a genetic change, but an epigenetic change that is causing genes to be silenced. And it happens frequently enough that we now call methylcytosine the fifth base in DNA. And it doesn't stop there. In the last 5 years, we know that this 5-methylcytosine can undergo further changes to other bases, which will also have influences on gene regulation. In the same way, the histone proteins the DNA is wrapped around are not static either. They undergo manifold changes. And these are all, the sum total of all these changes is that the proteins and DNA are interacting with one another with different affinities. And this is then determining whether a particular gene at a particular time in a particular cell is going to be turned on or off. And earlier I talked about a genetic code. In the same way, people speculate whether there's an epigenetic equivalent code, and there are hints that certain changes in DNA and histone proteins have certain biological consequences. So for example, like I said, when DNA gets methylated, that is a gene silencing effect. There are changes in histones. Some are associated with genes being turned on and off. But here you can see an example to show you how it's complicated. This particular signal can either activate or repress a gene, so it's not that simple. And if there really is an epigenetic code, it must be a very complicated language, and it's probably also going to be very cooperative and context dependent. So it's not going to have this clear code like I showed in the genetic code, where you have 64 words and each one of them has one defined meaning. And furthermore, if there is an epigenetic code, the number of possibilities is mind-boggling. You can see in this one histone protein itself, you have 16 sites that undergo methylation and acetylation, and that's only 2 of the changes. And there are literally about 100 different modifications you could think of in these proteins. So if you calculate these numbers out, you could literally have a Google of different states in the cell by these processes. And whether these are physiologically important, whether some of them are redundant, we don't know. But certainly the power of biology is immense in this regard. So how would epigenetic inheritance actually work? So you would assume that somebody is exposed to some environmental factor which causes them to have a phenotype, and this phenotype would then need to be transmitted to their reproductive cells, and then in their progeny it would be in this embryo state and somehow survive this reprogramming. And that's important because in humans and other animals, we do have these reprogramming steps, and I'll show you that in the next slide, where you wipe out all these most of these epigenetic signals anyhow. So it's very hard to see how much of this information you can pass over. But in principle, if you could, then the fetus and the adult would then have this phenotype that was transmitted through this epigenetic mechanism, and it could then perhaps be transmitted to its offspring as well. And this just reinforces what I said. So when you have these germ cells come together to create a new embryo, most of the epigenetic marks that they carry are wiped out. So you have a clean slate to create a new organism. And then the fetus gets bigger. And when the fetus then produces its reproductive cells, once again you get a clean slate of wiping out. So epigenetics, for it to occur, has to overcome these significant erasures. And at the moment, what we do know is out of about 18,000 genes, there are about 100 genes which do escape this type of erasing. And for these 100 genes, it matters whether the gene came from your mother or your father. So there are some genes where it's only the father's gene that's expressed, and there are some genes for which only the mother's is expressed. So somehow the embryo has a memory of this. And here's one example. So this is a gene that's expressed both maternally and paternally, so there's no discrimination. Here's a gene that in the brain is only coming from the father, and in the rest of the body does not discriminate much. Whereas here's another gene, where in the brain it comes from the mother, and in the rest of the body it comes from the father. So these are very subtle and difficult things to understand that we're only now starting to realize are happening in the genome. And here's another manifestation of epigenetics. So we all carry 23 chromosomes, 22 of them are 2 copies, whereas then we have the sex chromosome, which is the 2 X's in female and an X and a Y in male. And you know, in the Bible, you say the female was created from the male, but when you look at the chromosomes, it seems like female was the default, and the male was created just by adding a little bit extra to the female chromosome, okay? So anyhow, this is a big problem biologically for any genes on these chromosomes, on the X chromosome. So the female has 2 copies, so if the gene is on that chromosome, the female produces twice the amount of the male because the male has only 1 copy. So somehow this needs to be equalized, and this is done in different ways in different organisms. And the way it's done in females is that one of these X chromosomes is practically destroyed so that it's not expressing its genes. And this brings us back to the epigenetic cat. So if you have male cats, the situation is quite simple. The color comes from the X chromosome, so it either codes for black or yellow. And the male will just have 1 X, and if the X was orange, they'll be uniformly orange. Similarly, if the male was carrying the black gene, it will be uniformly black. With the females, now you have a choice because there's 2 X's. And if they both code for black, again, the cat will be black. If they both code for orange, it will be orange. These tortoiseshell examples happen when you have 1 copy of the black and orange each. And it turns out in the very early stages of embryogenesis, in females, one of these X's is being destroyed. And which one is being destroyed is completely random. So it's just a stochastic process, and that is why you see these patchy random patterns. So in some of these cells, it's the black gene that's being destroyed, so you express yellow, and vice versa. So if you see tortoiseshell cats, epigenetically, they are almost always female. You don't have to check physiologically. And how is this actually done? This is epigenetics. So this is chromosome-wide ablation. So a whole chromosome is being wiped out, and this is achieved at the molecular level by having a whole series of changes in the histone proteins and in the DNA. And this very complex process is going on in all the females in the room ever since birth. So that's a powerful example of epigenetics. And here's another interesting case of epigenetics that's quite well documented. On the right, you see a normal mouse, and there is 1 gene called the agouti gene, which is normally silenced in these mice because of DNA methylation, which is an epigenetic mark. So that's a normal mouse, and then there's this other gene phenotype called the viable yellow mouse, and these have a change in their epigenetics such that this gene is expressed throughout their life. And the phenotype is dramatically different. So these have a different color, they're much larger than the normal mouse, and they are more susceptible to disease. And you can do this, this was an interesting experiment that was done about 10 years ago. If you take a female yellow mouse that is pregnant and feed it a normal diet, then it mainly produces copies of itself which are yellow. However, if the mouse is fed a diet that is supplemented with methylation donors, that actually allows it to change the epigenome of the growing fetus, and the offspring that are produced are mainly brown. So there's been no change in genes, and in fact, this mouse is not seeing anything different after it's born, but just the fact that it was exposed to a different diet during pregnancy has caused this dramatic change. And of course, experiments like this have a lot of social implications too. And you could also ask the reverse question: what happens if you take pregnant females and feed them a substandard diet? Will that then cause problems? And although this might sound like an unethical discussion, this experiment has been done more than once, so we can see the results. And the most famous example was during the war, where for 1 winter, the Dutch were blockaded, and as a result, the diet was virtually starvation level, so the dropped to about 500 calories a day, whereas the norm is about 2,500. And what is noticed since then by following people who were conceived during that period is that, for example, if the mothers were exposed to the famine in mid to late pregnancy, then the babies that were born that were significantly lower in weight compared to normal. Whereas if the mothers were exposed to the famine in the early pregnancy, they were born with a normal weight, but then they had significant health problems later. And it's tempting to speculate that very early during that pregnancy period, they were programmed to adapt to starvation. So they are programmed to be eating because that's what they need to do in that time of hardship. But after they are born, there's no longer a famine, but because of this programming, these people continue to eat more than a normal person for the rest of their lives. And it is true that whether in early or late pregnancy, these babies that were conceived for the rest of their lives, which have been followed now for 6 decades, they have significant health problems compared to siblings who were born slightly earlier or later. And Audrey Hepburn is a famous example. She was conceived during this period, and throughout her adult life, she did have significant health issues as well. And similar observations have been made in other cases of famine as well. And so you can see these clear changes, and some of these changes are now being transmitted to the children of these children, so they're lasting for at least 2 generations. However, that's not truly epigenetics. To be truly epigenetics, you need to have these changes maintained for 3 or 4 generations. And we simply don't have the evidence yet whether this is going to be the case or not, so we'll have to wait and see. And those were all examples of epigenetics controlled by DNA changes, but this is not the only mechanism. So here's a review this year from Nature on RNA as a possible mechanism. So we are used to thinking of the sperm donating the genetic information from the father, the DNA, but the sperm is not just DNA. It contains various pieces of small RNA. These are not genetically coding for information, but they're probably influencing that early development. And it might be that this RNA is programming the epigenome as the embryo develops. And histone marks also can be transmitted. So this is a bit of a complex example in yeast that was reported this year in Science. And we are looking at one particular mark, which is the methylation of histones. And in this experiment, what they did was construct this system where when this methylation happens, it then leads to silencing of a gene, and that silencing leads to a red color of the colonies of fungi. And when you remove this signal, it reverts back to its normal type. Whereas in this bottom part here, what they did was delete one gene, which is a demethylase, and when you delete this one gene, you get this persistent methylation mark transmitted for over 50 generations of yeast. So this is not a genetic change. This is simply an epigenetic mark, and it can be persistently transmitted for generation after generation. So in the last few minutes, I'll give you some examples regarding practical applications of epigenetics, which don't have to do with whether this question of whether it's inheritable or not. And what is clear is that these epigenetic mechanisms, if they can be modulated, can also have a positive benefit in human disease. So for example, in cancer cells, you see many tumor suppressor pathways that would normally help the cell to be repairing itself. These are shut down in cancer cells. So does that mean that if we can inhibit this epigenetic mark, will that give you patient benefit? And it turns out that is the case. Similarly, in many cancer cells, some of these epigenetic players are highly expressed and working extra hard. And does that mean that if you stop that from happening, that could also provide patient benefit? And last but not least, some of these epigenetic proteins are mutated in certain subpopulations. And again, that suggests that if you can correct this defect, that might give you a cure for the disease. And this was the first epigenetic drug, which is more than 50 years old now, from Europe. And in fact, first in clinical trials 50 years ago. And this drug in particular inhibits the DNA methyltransferase, so it works by stopping DNA methylation. And it has been now used for a decade for the treatment of myelodysplastic syndrome, which is a syndrome that mainly occurs in elderly people and often progresses to leukemia. Histone deacetylase, so these are compounds that inhibit the deacetylase function of these histone deacetylase enzymes. And at the moment, there are 5 of them approved for various blood cancers. So there are patients taking these drugs. And with both these histone deacetylase and methyltransferase inhibitors, what happens is that the cancer cell, is given the opportunity to correct itself. So cancer cells are very artificial species. They do one thing very well, and that is divide over and over again at the expense of all the other possible fates they have. But with these drugs, the cell can then stop and either kill itself or repair itself or go down a different pathway and stop dividing. And here's one example of personalized medicine using epigenetics. So this is another of these histone marks. So histone 3, position 27, and this is methylated by something called the polycomb repressor complex to put on these 3 methyl groups. And this occurs in stages. So in normal humans, we have the wild type enzyme. So the enzyme carries out this conversion here, and that's fine. Whereas patients who have a mutation have a mutant version of the enzyme that once you are here, this will then go on to the second methylation and the third. And once you are here, this results in gene silencing that is, has a negative effect in cancer. And now that we understand this mechanism, it should be possible to have an inhibitor of this enzyme EZH2 and see if that can then stop this progression in these particular cancer patients. So this should not be a universal cancer drug, but one specifically selected for patients who have this mutation. And at the moment, there's more than one clinical trial undergoing using drugs that work selectively as inhibitors of EZH2. And these are very exciting developments for medicine, and we will see many more of these using epigenetic targets. Anyway, to finish, just some wrap-up points here. So the genome is hardwired. You can't really change it, and it's designed to be lifelong. Whereas the epigenome is constantly changing and evolving as the organism interacts with its environment. And it provides you with flexibility, which is what you need, because every one of us is being exposed to a different environment, and you need to be able to respond to that. And in some cases, yes, epigenetic changes can be transmitted to the next generation. But in animals, at least, the documented evidence for that, at least at this moment, is quite rare. And this is probably not going to be a major type of inheritance. But regardless of whether epigenetics is heritable or not, it's certainly true that modulating epigenetic pathways can be used for drug discovery. And there are drugs, as I've shown you now, in the clinic that work by this mechanism. So basically, you can thank or blame your parents and your earlier ancestors for the genome, and you cannot change your genome. But it's up to you to influence your phenotype through epigenetics. And you can either blame your ancestors and become obese, whereas I would say biology is more like playing poker. So you are dealt a set of cards, but you don't have to have the best hand to win the game. So it's all about what you do in your behavior. So in the same way, you may have genes that are good or bad. Some things you cannot change, but the epigenome is highly plastic, and what you do in every day of your life can have a positive or negative effect on your behavior. So I don't believe that epigenetics is this negative Lamarckian aspect where it's, but it actually has a lot of positive connotations for society. So with that, I thank you all for your attention, and I'll be happy to answer any questions.